This is going to be a fun lecture for me. I am a guy who has lived, breathed, dreamed, and partied CI, Cytoplasmic Incompatibility, for almost 10 years now. So, I wanted to give an extra lecture of CI, a cherry on top, okay? For the die-hard fans, the nerdiest of nerds, the few elite people who really want to understand the current state of cytoplasmic incompatibility research. And this lecture has to come now. It has to come around now because there has been an explosion of research. I could teach a whole class devoted to CI papers alone, and a lot of them have come out in the last 2 years. So I wanted to try to summarize the current state of the field as quickly and as succinctly as possible while giving you all the information you need to know to understand all the controversies, all the debates, all the fights about cytoplasmic incompatibility. And also, I wanted to have fun while doing it. So this lecture is going to be really fun for me. I can rant and rave about CI for hours upon end, okay? So I got my camera set up. Oh, and by the way, I am John Beckmann, Professor John Beckmann, Dr. John Beckmann, and I know CI. So let's walk on this trip together. All right, got the double camera, PowerPoint to record, not as a clutch. All right, I got the double camera, PowerPoint to record, not as a crutch. All right, let's pop a clog. Nietzsche says in your interactions, Nietzsche says in your actions, instead of giving people your hand, give them a claw. That's true, that's in Will to Power. Rare. Okay, so let's start here. At this paper, a Wolbachia deubiquitinating enzyme induces cytoplasmic incompatibility. Beckmann et al., Nature Microbiology. And let's also start here. Prophage Wo genes recapitulate and enhance Wolbachia-induced cytoplasmic incompatibility by LePage et al. In Nature. So we're starting here at these two papers. If you're not familiar with the previous lectures I gave on Wolbachia and CI, stop the recording, go back, watch the Wolbachia lecture, watch the CI introduction lecture, and then at that point we are ready to start at these two publications. So a nice way to tell this story is a story of two competing labs pushing each other to find the truth. I think that's a good way to phrase it. Let's be collegial and let's keep it at that. But the story is too fun not to tell. So actually, before we talk about these two papers, we actually have to talk about a Bordenstein paper which preceded these, which actually has some really important, interesting results. And that's actually this paper, Eukaryotic Association Module in phage Wo genomes from Wolbachia. So let me tell you the gist of this paper with respect to how and why it was important for CI research. So in this paper, they look at and they analyze the genomes of phages, which they can pull out from particles and they can sequence. And one of the interesting things that they reported was that when you sequence the phage genome, let's say here's an end, although it might be circular, here's an end over here. If you sequence the full genome, okay, one of the things that you see is there's this little section, okay? There's this little section which they call the EAM, the Eukaryotic Association Module. And in this module, within this genome of the phage, which they sequence from these particles, there are a few genes, more than a few, okay? There's some genes, and when you look at what these genes are and where they come from, they're not really phage originated genes, they are interesting bacterial genes, and some of these have eukaryotic motives, okay? So in some cases, they might not be bacterial genes. Some cases, they might have pulled things from eukaryotic genomes and assembled them into this little module that the phage carries. And one of the interesting things that was, I think, not a lot of people realized when this paper came out is that the CI genes, which we talk about a lot, the CIFs, the A and the B, those genes are inside of this eukaryotic association module. So even before we start talking about these two big papers, we have to kind of point out the fact that these two genes, which we were interested in from previously in 2013, which we identified in the sperm, go back, watch that lecture if you haven't seen it, these two genes, it appears that the phage is like carrying these two genes around. So this sort of discovery is important as an important pretext that sets up the conversation for when Seth's paper in the title you see prophage Woe genes recapitulate and enhance Wolbachia-induced cytoplasmic incompatibility. One of the ideas in this paper is that the genes that cause CI are from the Woe phage. Okay, and to really understand that, you have to understand the pretext of that EAM paper where they discovered that the A and the B genes pop up in this eukaryotic association module, and it appears that the phage carries them around. So this sets up the argument that Woe phage genes induce CI. And this is going to become a little bit important later because there are some debates about the importance of this and debates about where these genes originated from. And this is going to come into the discussion a little bit later. So I just wanted to give that pretext before now I go into the major points of the two big papers. And let me just reiterate here that my purpose is not to describe in full detail all the data from all these papers that I'm going to talk about, okay? My idea is to synthesize why these papers were really important to me, what I saw as important pieces of data, of the infrastructure that sort of precipitated the understanding of the molecular biology of CI. So some people might get upset if I talk about their paper and I just skim over some other things. That's just what I'm going to do, so you'll have to deal with it. You can slap this like if you want to do that. So let's start with the major points from my paper. Again, hit pause if you haven't seen the last lecture. Go back, watch that lecture where I describe in quite a bit of detail the data of this paper. But I'm just going to skim and write out kind of the basic points of this Beckmann et al. Nature micro paper. And this is in 2017. So this paper starts upon the premise that these operons that we're studying are constructed as toxin antidote systems. Okay? And one of the first ways that we could biochemically test whether or not these were toxin antidote systems is by testing binding interactions. Because type 2 toxin antidote systems, which are constituted of two proteins, a protein that's an antidote, a protein that's a toxin. In those systems, usually the antidote behaves by binding the toxin. So one way we could start to sort of address is this operon a toxin antidote system is to test the binding, okay? And we did that. That is this data right here. So if you look at this figure, we're overexpressing and we're checking for binding of the red B with the red A, and you can see by this Western blot that it binds. Here we're checking the black A or the black B with the black A, and they bind. So this data is the data that proves, at least in some sense, these two proteins, when they get made, they're binding and they're interacting, okay? And that supports a toxin antidote hypothesis. Okay, 2, and I'm going over this quickly because I went over this in detail in the last lecture. The second way that we can test, is this a toxin antidote system, is let's express these proteins in yeast, and if the B is a toxin, the B should kill the yeast, okay? And that's these data, these data. That is these data right here. You see that if we overexpress the B proteins, it kills the yeast, okay? So that supports, again, supports the idea that this is a toxin antidote system. So let's label point 1, big impact of this paper is binding. We'll call this cognate binding. And let's label 2, big impact of this paper, toxicity plus rescue in yeast. Okay, so let me show you the rescue experiment. If it's a toxin antidote system, if we express the B protein with its cognate partner A protein, we should see rescue. We should see growth, and that's what you see right here. Whereas if it's mixed with the wrong A protein or no A protein, there's complete death. Okay, so that's what we see. So that again supports the idea that this is a toxin antidote system. Now, the next big thing that this paper did, which I didn't talk about a lot in the last paper, was it describes the biochemistry of how these genes function, okay? And it posits that the B protein, SIDB, is a D-ubiquitinase. Okay, so just for a brief overview of this, okay, ubiquitin is a post-translational not large, but small peptide that gets post-translationally modified and added to substrate proteins. Okay, and ubiquitin is important for regulating degradation of whatever this substrate is, also localization of this substrate, molecular functions of the substrate. Ubiquitin can change things once it gets attached, okay? And a D-ubiquitinase is an enzyme that cuts this bond and releases ubiquitin from the substrate. So that's what a dub does. So we discovered that SIDB, this enzyme that causes CI, is a D-ubiquitinase. So let's look at the data for that. One of the data that's important is this thing right here, SIDB star. So if you look at the protein SIDB, you see this domain here we annotate as ULP1 protease. Okay? That is the same thing as a dub. It's highlighted here. I don't know why I did that. Very strange. That's the same thing as a dub, okay? And we know how to inactivate those dubs. We make what's called a catalytic mutation, which is we change the core cysteine, which catalyzes this reaction that I just drew. The cysteine, an amino acid at the active site, is responsible for that activity. So if we mutate the cysteine to an alanine, we should be able to inactivate that activity, okay? So that is these data right here. So do you see how SIDB by itself kills the yeast, but if you inactivate the dub through mutation, the yeast are perfectly fine. Therefore, we conclude that this toxicity, the toxicity from that B enzyme, is due to the deubiquitinating domain. Now that's all fine and dandy in yeast, but we need to prove it biochemically that it is a deubiquitinase. So we do those experiments. That's these experiments right here. I think this is figure 3, okay? And what I want to go over is just this figure right here, because it'll give you the basic gist that this is a dub. So ubiquitin is a peptide, okay? And it forms what are called ubiquitin chains, okay? So this is a test where you're seeing an anti-ubiquitin Western blot. So we're using an antibody that recognizes ubiquitin, and we're loading in ubiquitin chains. So you see how down here would be a monomer, one ubiquitin, but there's nothing there. Here's diubiquitin. That's 2 chains, okay? And then this right here is 3 chains, 4 chains, 5 chains, 6, did I get that 2? Oh, 3, 4, 5, 6, and sometimes up there you can see a 7, okay? But these are chains of lysine 48 linked to ubiquitin. And if SIDB is a dub, what it can do is it can take those chains and it can cut them up, cut, into a monomer, which appears down here, okay? So when you mix the ubiquitin chains with SIDB, all of a sudden this band appears. That is the monomer. That proves that CIDB is a dub. If you inactivate the catalytic cysteine, that band disappears. So you know CIDB is a dub domain. Okay, so we know how CIDB functions. We know how it's inducing its toxicity. Point 4 is transgenic expression of CID-A plus CID-B induces synthetic CI. And these are the data for that. Here on the y-axis is progeny, so offspring of a cross, okay? And 30 crosses, n equals 30 crosses. And what we're doing is we're crossing a male that has the CID-AB operant expressed to a female that's wild type, and she produces no offspring. So we get complete sterility with overexpression of the CID-AB transgenes. Point 5 of this paper is the cytology of the synthetic CI mimics CI cytology. Okay? And these are the data for that. Okay? So, how do I explain this? So there's a very classical phenotype when you're looking at CI cytology, and this is described in detail by many, many authors, and the best pictures of it are by Bill Sullivan's lab. And what you see is when you look at CI crosses, the chromosomes that derive from the father at anaphase right here, when the cell's trying to pull apart the chromosomes from the father are not condensed properly, and they don't pull apart, but the chromosomes from the mother do pull apart, okay? So the important point is that this is not just a random sterility effect. The cytology mimics precisely the CI cytology, the wild phenotype that you would expect to see were you to have true CI. So those are the major points of this paper. We conclude CID-A and CID-B are responsible for CI, and CID-B is the toxin, and CID-A is the antidote. Now I'm going to go into the nitty-gritty of the questions that I know are popping up into your mind right now. But first let me go and talk about the other paper. Now Seth will not like me paraphrasing his paper, okay? But as I said, this is a lecture of what I think were important to me in my perspective on this field. So let's talk about it. And if you didn't notice, I'm also an author on this paper, so it's technically mine too. So let's talk about it. Now, before I start to talk about this figure and why I think it's important, let me just premise the fact that we already knew which genes at this point were causing CI. Okay, we already knew that because in 2013, again, if you look in the past lectures, I explained this in detail, we knew that these genes were being overexpressed in sperm, and they were the only proteins from Wolbachia that were being found in the sperm. So we knew that these proteins were involved with CI, okay? But we had to sort of convince the community, and Seth had to sort of convince the community what the good case that these are the best targets for CI. So I do think that this figure is important, and I do think it contributes a lot. So what this is a big genomic comparison. So in brief detail, we've talked in the past about bi-directional incompatibility, okay? So this is where you have two different Wolbachias. Let's say you have a red Wolbachia and a black Wolbachia, okay? And when these different insects that harbor them mate, you can get CI. And that's because the CI systems don't match properly. So one way you could go about discovering what are the genes that are important for CI is you get a bunch of these different strains of Wolbachia that are all mutually bidirectionally incompatible, okay? And you sequence their genomes, and then you can do essentially like a large blast analysis. You can take all the genes from let's say Wolbachia 1, and you blast it against all the genes from Wolbachia 2, and you blast it against all the genes from Wolbachia 3. Okay, and what you're looking for, if they're bidirectionally incompatible, what you're looking for are the differences. So genes that are different fit in here. Okay? Now you might be asking yourself, Beckmann, why are you on this paper? Why are you on this paper? Well, let me tell you. At this time, when you blasted the genomes of the bidirectionally incompatible Wolbachia that we had, this list that was in this category that was mutually different between all the incompatible strains, that list was about 15 genes, okay? That's kind of a lot. That's a lot of genes. You still don't know which are the genes that are causing CI. So why am I on the paper? You see this right here? This is a proteome in the ovaries. Let me explain that. Let's go back to this. Let's say there's 15 genes that are different between all the different incompatible strains. So your list of candidates is up to 15. How can you reduce that even more? Well, you can make the hypothesis that whatever's causing CI or rescuing CI, specifically rescue, has to be expressed in ovaries. And you can figure out all the Wolbachia proteins that are expressed in ovaries by doing a proteome, which I did. So I contribute the proteome, and when you add the proteome into these comparative data sets, now what you're looking at, all the genes that are different between all three incompatible strains plus the category that they are expressed in the ovaries. Once you do that, then it becomes beautiful. You get 2 genes. Guess what those genes are? SID-A, SID-B. So this big first figure is pointing out that the only 2 genes that could explain the different bidirectional incompatibilities are SID-A and SID-B. Now let me summarize this part. This part is essentially telling you there's different types, okay? Different types of CI operons. So let's make the list of important points to take home from the LePage et al. Nature Pub. 1. Genomic comparisons point to SID-A, SID-B operons. 2. There are different types. Now here's where we start to see some differences between the 2 groups. And I'm going to talk more about the differences, but let me put a pause on that. Okay, we'll come back to that. Point 3 is transgenic overexpression of SID-A and SID-B induces CI. Let's look at that data. That's this data right here. So what you're looking at on this x-axis is what's called a hatch rate. Oh, here at 100%, there's 100% of the embryos surviving. And here at 0%, all the embryos are dying. So you're getting killing effects, which is CI, by transgenic overexpression of the 2 genes together. Now the 4th important point of this paper's contribution is a W-mel infection. So this is Wolbachia from Drosophila melanogaster. That infection rescues transgenic CI. Transgenic or synthetic. I prefer transgenic, but sometimes I think I say synthetic. And these are the data for that. So you go from this hatch rate to almost 100%. That's the rescue experiment that shows you that an infected female can rescue the transgenic effect in the male. Okay, so we got these two simultaneous papers. One's a little bit more focused on the biochemistry. One's a little bit more focused on, I guess, the genomic comparisons. But let's talk about where they agree. Where did we all agree at this point? At this point, we all agree that these two genes, A and B, are the factors that cause CI. We all agree on this major point, okay? Now let's talk about the differences, which is where the fun comes. And the first major one of these differences, I will call it by its name, will be called the 2-by-1 model. Now in this paper they don't call it that, okay? But this is where you're starting to see the emergence of this idea, okay? And the 2-by-1 idea is that B is not sufficient for induction of CI. That's one of the roots of 2-by-1. And the idea is that the A protein, when it gets made, the A protein somehow helps B to induce CI. Now, the fact that the 2-by-1 model is starting to emerge now is shown by the one word which they add to the title, which is end enhance. Okay, so let's talk about that data. So wild CI in the laboratory, you can kind of tweak it. You can tweak it with different things. You can age males to tweak the CI hatch rate levels. You can change temperature in certain contexts to tweak CI levels. So what you're looking at here is sort of like a weakened CI. That's this line right here. So here's like a weakened CI. If you overexpress this gene 631, which is the A protein, that CI gets worse, okay? And if you overexpress the B protein, the CI gets worse. And if you overexpress both, it gets even worse, okay? So these are the data that are starting to show or to support the emergence of the 2-by-1 hypothesis, where the idea that A helps B, okay? Now, if you carefully read the discussion in here, they make a mistake, which gets corrected later in the literature. And what they posit in the discussion is that the A protein is probably a second toxin. And the rescue factor, importantly, the rescue factor is unknown. Now let's compare that with the discussion and the conclusions from the Nature Microbiology paper, the other paper. That paper shows rescue by A in yeast. Look here, SID-B plus SID-A rescues the phenotype. SYN-B plus SYN-A rescues the phenotype. So in a disagreement, we have two models emerging. A TA model, where there's a toxin and an antidote, and a 2-by-1 model, where there's two toxins and we don't know the antidote. Now, let me just carefully say, this is not the current state of the 2-by-1 model. This was the state of the 2-by-1 model at the point at which these Nature Microbiology and Nature publications came out. Now, there's some other data that I would point out, which you would not have seen unless you very, very carefully read the papers, okay? In the Nature Microbiology paper, arguing for the TA model, we tried the experiment where you just express SID-B alone, okay? And to do that, you have to make a transgenic insect to express SID-B, okay? So we made those constructs, and we put them into a fruit fly, and what happened was all the fruit flies immediately died. They died. So we were not able to ever recover a transgenic insert of SID-B. Now, in our opinion, that was evidence that B is a toxin. Because the only way you can get it into the genome is by complexing it with its SID-A partner. So we were able to produce a transgenic insect that had both SID-A and SID-B. And our rationale was, well, we can't get SID-B alone, but we can get both SID-A and SID-B. That suggests that A is sort of alleviating some of the toxicity of SID-B. Therefore, that supports the toxin antidote model. Now, these data are directly in contradiction with some data in the Bordenstein paper. In the Bordenstein paper, they do get insertion of SID-B alone. And that's right here. And they saw no effect. Therefore, their conclusion is, again, 2x1 model. B protein is insufficient for CI. You also need the A. Now, I will just say these data are not completely contradictory in a sense that we were working on SID-B WPIP. This is from Wolbachia that lives inside of Culex pipiens mosquitoes. The Bordenstein paper is working on SID-B WMEL. Okay, this is Wolbachia from Drosophila melanogaster. So this is a completely different ortholog of this gene. There's the same gene with a little bit of a few different SNPs. Okay, there's some differences in amino acids which are not insignificant. Okay, so we're not working on exactly the same protein, but we're each sort of observing some subtle differences here, and that sort of started to precipitate a difference in the models, where you have kind of a thinking of a TA versus a 2x1 model. But I don't like to see it as versus, because in my view, and as you'll see in the future of this lecture, these models are not incompatible. Do you like my joke? These models are not mutually exclusive. They could both be true. So let's talk about that more. And let's also derive another big difference between the two, okay? A second difference, B, was nomenclature. From the Nature papers perspective, the Bordenstein perspective, they didn't have data to conclude that the SID-B gene was a dub, because that was in our paper, although Seth was aware of that data. But that's all I'll say. Seth's conclusion was, we don't know how CI works, so let's call these genes factors, CIFs, CI-inducing factors. Now, from our perspective, we didn't know how they work. It's a dub. SID-B is a dub. So we felt biochemically it would be important to include the biochemical nature of this enzyme into the name. So our version was CI-inducing deubiquitolase. That's where SID comes from. Okay? Now, this nomenclature also relates to a third difference between the papers. Okay? And that's in discussion of the different types. Seth's paper goes into elaborate detail outlining the different types, type 1, type 2, type 3, type 4, and these come from different evolutionary tracks with a common ancestor. Our view was not necessarily in disagreement with the evolutionary history of these different types, but that the most core simple thing that made some of these paralogs different was that some were nucleases and some were dubs. So if you were to look in our paper at this figure right here, we're starting to introduce the idea that there's many different paralogs of these, and sometimes in QLEX you find multiple copies, okay? Multiple different paralogous and homologous copies of these different operons, and some of them have this dub protease, and some of them have this nuclease, okay? And so we differentiated these operons based on biochemistry, because we felt like that was helpful. In the lab, when we were discussing these, we were either working on CINs, CI-inducing nucleases, or DUBs, CI-inducing deubiquitinases. And so it's very quick and easy for us to say, well, we're talking about the CIN here, we're talking about the CID here, okay? Because we're focused on biochemical experiments. So that's where the sort of the different nomenclature, the different ideas of the nomenclature comes from. Now at this time, when we're thinking TA versus 2x1, when these two groups are thinking about this sort of dichotomy or this different modular systems, another piece of evidence that was in support, I guess, of the 2x1 model, depending on how you look at it, was the fact that when we took CID-B WPIP and crossed that to a female that had CID-A WPIP, we could not get rescue. We could not show rescue, okay? And that was one big piece of data that would have been at the time in support of the 2x1 model, that the A gene was not rescuing. And if you remember, at this time, like I just said, in the Nature publication, they posit that the A protein is not, in fact, an antidote. And we don't know the antidote, whereas we were positing it was the antidote. But we couldn't get the rescue. And our rationale for why we couldn't get the rescue is, we thought, well, in our system, these embryos are completely dead. Look at that data. It's literally zero. They're completely dead. They're so dead, in fact, that they might not be able to be rescued. In effect, by transgenic overexpression of this operon, we might have crushed the embryos so much that it would be impossible to rescue them. And we posited if there was some way, and we said this to the reviewers, we said this to the reviewers, if there was some way where we could increase the overexpression of CID-A and get more of it around in the female, we might be able to produce that rescue experiment, okay? So it's important that you know that context, because there's some funny things that come out later. And so the end result was that the reviewers completely ignored the fact that we had biochemistry. They didn't care about it. They rejected our paper because we did not have rescue, and they accepted the other paper because it did have rescue, which I think was a gigantic mistake, and those reviewers are complete morons. And we all agree now that they were wrong. So what happened next? Well, I presented this data at the Wolbachia meeting in Australia. And after this presentation, I distinctly remember, it was the most shocking experience of my life. I literally got 30 to 40 minutes worth of questions, a grilling, a second thesis defense on this research, because nobody wanted to believe the results. And I remember one question specifically, that they asked me is, how do you explain QLEX? Now let me give you the context for that question. Remember when I talked about Levin, and remember when I talked about how Levin discovered CI in the QLEX systems, okay? And what Levin did is he collected a bunch of different populations of mosquitoes from Europe, and he crossed them all together and formed this big, large genetic analysis, or maybe not genetic analysis, but crossing experiments, and determined the different cytotypes. And what he came up with was that there's approximately 17 different, essentially, CI systems. There's essentially 17 different cytotypes that are incompatible with each other. How do you explain that? How do you explain with two genes, CID-A and CID-B, how do you explain that? There needs to be at least 17 different copies, in theory. And I said, at the time, I literally said this at the time. I said, oh, that's easy to explain. There's just genetic duplications of the operon. And we see that in multiple cases, especially in QLEX. I've cloned at least 3. That was my literal answer. There's multiple different copies, okay? And if there's multiple different copies of SID-A and SID-B, they can just be duplicated and be evolving and easily evolve these 17 different cytotypes of the QLEX system. And then boom, Manot's paper drops. I think I'm pronouncing it correctly, Manot, and it's Mylene Wheels Group in France, in collaboration with Matthew Sicard. So boom, this paper drops, and what does it say? It says, we show here that the remarkable diversity of CI in the QLEX pipiens complex is due to the presence in all tested WPIP genomes of several copies of the SID-A SID-B operon, which undergoes diversification through recombination events. So in this paper, what they did, PCR amplified the different operons of SID-A and SID-B from these different cytotypes of QLEX, and they discovered that, boom, there were multiple copies. They were being duplicated and evolving. Thus, the explanation for the 17 different cytotypes was due to the SID-A, SID-B operon duplications. Not only that, the other reason why I loved this paper is because they point out that these 2 genes are co-evolving, which is consistent with the toxin antidote system in which the gene B co-diversifies with the gene SID-A. And one of the only factors that could explain some of the phenotypic rescue events was SID-A. And in a follow-up paper, they specifically described that the SID-B gene is associated with the modification, or in other words, the induction phenotype in QLEX pipiens. But SID-A is not associated with that. Therefore, these papers argued in support of a TA hypothesis and did not support the idea posited in the 2x1 at its current state that the A protein was a secondary toxin. So I'm talking about this story as it's happening, as we're looking at the hypotheses, as data is emerging. These are the current pictures. And the current pictures were A is an antidote, B is a toxin, or the other idea was no, A is not an antidote. 2x1. A is a second toxin. Then boom, this preprint dropped. This is the publication, but the preprint for this dropped. So let me explain some of the important data in this paper, which is Brandon Cooper's lab in collaboration with Michael Torelli. Megan is the first author. Let me explain the important data from this paper. And the title is Loss of Cytoplasmic Incompatibility and Minimal Fecundity Effects Explain Relatively Low Wolbachia Frequencies in Drosophila mauritania. This paper was hugely important, okay? This preprint was hugely important because there was a crucial genome that we were missing at the time. And it was this genome of this very, very unique Wolbachia, which was WMA, or in some cases you see it WMAU. Now, why was this Wolbachia unique? This Wolbachia was unique because this Wolbachia was one of the few described rescue-only strains. Now, what does that mean? That means if you take a male with infected, this is a fly, with WMA, and you cross it to something else, a female, let's say a female uninfected, there's no CI. But if you take, and I hope I'm getting this right, if you take a male fly that has a different bidirectionally incompatible strain called W-no, and you cross that to a fly that has WMA, Wolbachia mauritania, this female can rescue that CI. So it can rescue CI, but it cannot induce it. So what would you expect? What would you logically expect if the toxin antidote operon hypothesis is true? What would you expect to see in the genome of CYD-A and CYD-B from Wolbachia, from Drosophila MA? The answer is that you would expect to see something messed up in the CYD-B toxin. Therefore, it cannot induce CI, but you would expect to see the CYD-A antidote intact. And that's exactly what they describe in that paper, okay? There's a gene disruption, a mutation, that causes this gene to become pseudogenized. That means the gene has been destroyed. So there's no toxin, there's no more a functional toxin, but there is a functional antidote. So this paper was, again, another support for the TA model. Okay, so what comes next? And this is, again, this next argument can again be framed within the TA versus 2x1 models, okay? One of the things that we pointed out in the 2013 paper was that these two genes are an operon. Okay, that means they're polycistronically transcribed. That means the RNA polymerase pops on at the promoter and it transcribes a message that codes for both genes. And then when the ribosome feeds in this message, it produces two separate proteins, an A and a B. That's an operon. Polycistronic transcription. That means there's two proteins, in this case, encoded in one messenger RNA molecule. Okay? Now, this is important because in bacteria, many toxin antidote systems are organized as operons. And in the original 2013 paper, which I described in a past lecture, what we did was we looked at these operons and we did RT-PCR. So that's PCR of the message. Now, I'm quickly skipping out explaining cDNA here, but we did a PCR of the message with a primer binding here and a primer binding here. And if we were able to amplify this product that spanned the intergenic gap, that equals proof for polycistronic transcription and therefore proof of operon status. So then this preprint drops from now Professor Amelia Lindsay and Dr. Irene Newton. Amelia Lindsay is now a professor at University of Minnesota. So I have the utmost respect for Amelia Lindsay because she comes from my alma mater now. And the University of Minnesota has a great history of Wolbachia research because Hertig, who discovered Wolbachia, did his PhD thesis on Nosema at the University of Minnesota and then went on to discover Wolbachia. And me, the great me, discovered the CI operons at the University of Minnesota. So I have all the utmost respect for Amelia Lindsay, and I hope she serves the University of Minnesota well. But this preprint pissed me off. Because in this preprint, they do RNA-seq of the two genes. You have the A and the B gene. And you look at the quantity of the messenger RNAs produced. There's more A and less B, okay? So what they say is, if this was really an operon, you would expect to see a polycistronic message, and therefore there would be the same amount of A as B, 1-to-1. But what they observe in the data is more of A and less of B. And they argue that due to this, due to these data, it's not an operon. Therefore, TA is unsupported. Now I'm going to tell you, it doesn't matter. It doesn't matter if you have more of A and less of B. It could even be opposite of that. I had issues with this, okay? I had issues with this because I can literally draw for you a case where you get this phenotype, okay? And it's still an operon. So let me draw a system here. Here's a promoter. Here's the A gene, A gene. Here's the B gene, B gene. Now let me show you a scenario. RNA polymerase, which is in green, comes on here at the promoter and it starts transcribing A. Boom. Falls off. And this can sometimes be induced by sequence in the gap. Now what happens if you do this a lot? Okay, let's do this again. RNA polymerase pops on, boom. Falls off. RNA polymerase pops on a third time. Boom. This time it made it. It made it! Smiley face. It made it all the way to the end. Now, if this were to happen and you had a special sequence, you could literally program different stoichiometries. I'm going to spell this completely wrong, but stoichiometries. By programming with a probability that the RNA polymerase falls off. In this case, you're going to get 3:1 A to B ratios. Okay? Now, their argument was this proves it's not an operon. If there's differences, they should be exactly the same. But I've just drawn for you a scenario where you can produce A to B in different ratios, and it's still an operon. And I had funny exchanges with Dr. Newton's laboratory about this preprint. And I don't recall completely the exact conversation, but I think I said something like, Irene, please just PCR the operon. Please just do the RT-PCR experiment. Design a primer right here, design a primer right here, and I can guarantee you, you're going to replicate my results, okay? So Irene did that. So here, look at this data right here. These data. These data. These data. Look at these data. Okay, so let me explain what this is. If you have Sid A, WMel, Sid B, WMel, and you design a primer here and here, which is going to recognize the transcript. If it's an operon, you should be able to amplify an amplicon that corresponds to the entire message that goes across the gap. That's what this is! She reproduced my experimental results. But of course, everybody had already read the preprint. Nobody goes back and reads the final publication. So the preprint was what everybody remembered. Nobody remembered that her data actually supports my conclusion that it's a toxin antidote operon. So in the discussion, now I'm paraphrasing here, in the discussion, there's something written like, well, it's not an operon. So she's trying to say it might actually be an operon. It might be an operon. Therefore, I would say TA is supported. Oh my God, I gotta pop another White Claw. True story. If you ever discover something important, nobody will believe you for at least the first 5 years. So when I presented this at the Wolbachia meeting, Scott O'Neill came up to me, and I don't know what the purpose of the conversation was. I was not in a mood to hear it because I had essentially had my paper rejected, and I presumed it was Scott O'Neill. I don't know if it is or not. I presumed it was him. And I got nothing against him, but he came up to me And he was like, you know, you got to just relax a little bit. You got to chill out. And shit, what did he say? He said, quote, when you discover something like this, it takes a long time for the field to catch up. What you need to do is you need to keep putting out the data and let the data speak for you. Don't be talking like you're talking. I was not in the mood to hear that. But that's what he said. Probably actually really good advice. So thanks, Scott. Cheers. That said, I do my own thing and I do it my way, and that's the way that I do it. So let's continue. Okay, so at this point, like, I felt like there was so much data supporting the toxin antidote hypothesis. I thought like it was insane for people to not believe in the toxin antidote hypothesis. But people were set on the idea that A plus B were inducers. By that I mean two separate toxins, both were toxins, and that the rescue factor was unknown, okay? Despite all the data that I just showed you, all the data that I talked about supporting toxin antidote hypothesis, people were set on the idea of the 2x1, that both of these genes are toxins and the rescue factor is unknown. Even Michael Torelli. Okay, so I had a conversation with Michael Torelli. His lab at some point read both these papers, okay? They digested both the papers, and after he had digested the papers, we had started an email conversation. And we were talking about toxin antidote hypothesis. And I remember distinctly, Torelli said verbatim, well, toxin antidote hypothesis is a nice hypothesis. It would be perfect if it made sense. But it's not true. Look at Seth's data. And I remember thinking, what the fuck? What the hell is this person thinking? Like, there's so much data supporting toxin antidote. And there was nothing I could do to explain that, like, there's all this data supporting toxin antidote hypothesis, okay? So it was baffling to me. And all I remember saying was, well, the yeast data for sure support toxin antidote hypothesis. And I am not willing at this point, when we know almost nothing about the CI system, I am not willing to throw away my yeast data to onboard Seth's 2x1 idea. And I specifically said at the end of that conversation, and I said specifically, you just wait. I said this to Torelli. You wait, and I will prove it. I will prove that toxin antidote hypothesis is correct. And I will do it by proving that the A is the rescue factor. And then in the sickest, most deformed twist of fate, Dylan Shropshire, Seth's lab proves that the A protein is the antidote. That's this paper right here, this PNAS paper by Dylan Shropshire. And I got no beef with Dylan Shropshire. I got a fun story about Dylan Shropshire. But this paper was good. I can't lie. This paper was good because it proved my fucking hypothesis. But it was sick that he got it. It was like, oh. Sick twist of fate. One prophage Wo gene rescues cytoplasmic incompatibility in Drosophila melanogaster. So let me describe what they did. Remember back when I said to the reviewers, if we could only just express CYT-A in a higher amount, we would get rescue. We would be able to produce the rescue experiment if we could just increase the expression of CYT-A. Well, what did Shropshire do? They switched the driver. So we were previously using this driver called NGT. So these are drivers. These are transcription factors that turn on transgenes. okay? And you can get drivers of variable strength. So they switched to what's called the maternal triple driver. This is a line developed by Lynn Cooley at Yale, okay? And what they did is they have inserted multiple Gal4 transcription factor inserts on each of the chromosomes of Drosophila and made them all homozygous. It's a triple homozygote, so it's called the maternal triple driver. It's like superpowered expression. When they do the maternal triple driver, CYT-A, and they cross the CI male, cross to the MTD maternal triple driver CYT-A, they get rescue. So Shropshire proved it. Shropshire proved that the A protein, A, is an antidote. Okay, this is a positive step. So let's take a moment here to just discuss the positivity of this result. Because now the two groups, TA versus 2x1. Now the two groups are in agreement. The two groups are in agreement. I probably spelled that wrong. The two groups are in agreement that the A protein is the rescue factor. Okay? And now's when you start to see the more terminal form logic, the more terminal logic of the 2x1 pop out. Where instead of before, what they were positing was that A and B are both toxins, rescue factor is unknown. Now what they're positing is A plus B are necessary for induction, plus A is antidote. Okay, and we are in agreement that A is antidote. Both TA and 2x1 are in agreement that the A protein is an antidote. So finally, thank you Dylan Shropshire. Thank you, Dylan Shropshire, for proving my hypothesis that the A protein was an antidote, for validating my yeast data, which I originally first showed rescue experiments in the yeast. Thank you so much for validating my hypothesis. You have my eternal gratitude. You want to hear my funny story about Shropshire? Okay, here's my funny story about Shropshire. I visited Seth's lab, and I had a meeting with Dylan. And again, I like Dylan. I thought he was smart, okay? So I met with Dylan, and we were talking about his thesis experiments. We were talking about his thesis. And I don't remember the particular experiments. I don't remember the particular context. But he was saying, this is what I'm working on. And I said, oh, that's a cool project. You should consider doing this experiment. And I laid out this experiment, blah, 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 do this and that. And he said verbatim, oh, like I thought of that too, but Seth told me not to do it. Seth told me not to do that experiment. And I said, quote, you need to stop listening to Seth Bordenstein. So Shropshire, mad props. Thank you for producing this fantastic data. Okay. So now as the hypotheses are evolving, we have TA versus, we're starting to see an evolution of the thinking, the logic behind 2x1. And here's where I think we can pause and we can start to talk about the agreement. And so let me just talk about how I see these hypotheses, okay? I see the toxin antidote idea as the idea that there is A as antidote and B as toxin, okay? And I see 2x1 as it stands right now, a subset idea within the toxin antidote framework. Because as I described above, we all now agree that A is antidote. We all agree on that, okay? So we're in agreement. What we are not sure about, what we do have disagreement about, is the two-part, okay? So the one means A rescues, now, according to their most recent publications. The two-part now means, as I have written before, A plus B are necessary for CI induction. And I disagree with that for multiple reasons. One, you can express B proteins in yeast and you get toxicity without A. Two, and that's this paper by my great friend and colleague, Hongli Chen, I'm on this paper with also my other great friend, Judy Rano, and our great mentor, our great leader, Mark Hochstrasser. So this is our PNAS answer paper, which is a Wolbachia nuclease and its binding partner provide a distinct mechanism for cytoplasmic incompatibility. So let me talk about some of the interesting data from this paper, which is relevant to the 2x1. But first, let me summarize what this paper was about, okay? So when we originally put out the two papers, remember we were talking about the different types of the CI operons, and we had this set up, this dichotomy, which we felt was a biochemical dichotomy, where we had some that were SIDs, dubs, and some that were SINs, nucleases. And Seth's disagreement with the idea of the SIN and the SID was that it's premature to call these things a dub or a nuclease because A, you haven't proved that it's a nuclease yet, so nuclease question mark, and B, we think there's other things that are important for CI, not just the dub. So let's call these things factors. Okay, so our answer to that was, well, we've already studied the dub. We've already shown that the dub is necessary for CI. We've already shown that it is a dub. Therefore, we can call it a dub. Now let's study the nucleases. Okay, so that sets up the pretext for Hung Lee's beautiful paper. Now, evolutionarily, what are these nucleases? Well, we knew that the SIDs and the SINs were related, okay? Because if you blast these, you see homology here. And what's different about them is the SINs lacked the C-terminal domain, which had the dub. And the SIDs had a homology to a nuclease domain, but they lacked the catalytic residues that would actually cut DNA, okay? So we always thought the SIDs are dubs, the SINs are nucleases, because unlike the SIDs, the SINs had an active nuclease, okay? So that was our hypothesis. So this paper has multiple important points. One, the SIN is homologous to other nucleases, and you see this DEK, the black residues, DEK, that is the catalytic site. It's got two conserved nuclease domains. Therefore, we hypothesize that this protein is cutting DNA. Now, the easiest way to test that is to literally just purify plasmid and mix it with purified SYN-B and see if you get digestion of the plasmid. That's the easiest assay for nuclease activity. So that's what this figure is here. Look at this data right here. This is just circular DNA, that's plasmid DNA. So here are the plasmid bands. If you add SYN-B plus DNA, it disappears. It's gone. It cuts it. It is a nuclease. If you add EDTA, which chelates divalent ions, which are necessary for nuclease activity, you no longer get the cleavage. So it's definitely a nuclease. You can inhibit it by adding EDTA, which pulls out the divalent ions. You can also inhibit it by mutating the catalytic site of the nuclease. So here they mutate the lysine to an alanine, which is key for cutting the DNA, and it no longer cuts the DNA because you can still see DNA bands. So SYN-B is in fact a nuclease. Therefore, remember when you told us that we couldn't call it a SYN because we hadn't proved that it's a nuclease yet? Well, it is a nuclease, so we can't call it a SYN. Boom, game, set, match. Now let's go back to the 2x1 hypothesis, okay? There's some really interesting data here that sheds some light on 2x1 versus TA. Or if they're not mutually incompatible, which I don't think they are, shed some light on the discrepancies between the two, the nuances. Look at this cross right here. So these are hatch rates again. 100% means fully alive, 0% means completely dead. Look at this cross. Here's overexpression of B protein by itself. Look at that hatch rate. It's about 65%. That's a decent strength of CI with just SYN-B alone. So SYN-B alone is sufficient for CI. Therefore, does that not immediately disprove the 2-by-1 hypothesis? You answer the question. But it does get more complicated. It does get more interesting, okay? So if you mix Syn A with Syn B, it gets worse. Wow, so these data are complicated. Okay, so let's talk about that. We got TA versus 2-by-1. In Hong Lee's paper, Syn B alone gives CI. That supports TA. That B is toxin by itself. But A plus B gives stronger CI. That kind of supports 2-by-1. I'm not going to lie. It kind of supports 2-by-1. But this disproves 2-by-1. So, like, what do we think? What's the deal? So, I want to tell you what I think the deal is. I do want to tell you what I think the deal is. But to do that, I have to introduce another paper by Joe Gillespie, that handsome devil. But even before I talk about that, I have to bring back the EAM story. Remember the EAM story? I have to bring that back because we have this track of CI mechanism research, which I've been talking about this whole time going forward on this track. And what we have with Joe Gillespie is we have this track of Rickettsia plasmids research tracking closer and closer and closer to CI. And there's a point at which these two research tracks meet. Okay, so let me start here and let me start here. So even before I start that, let me just say that there's a clade of bacteria, and within that clade are Rickettsia, I'm going to get this tree wrong, Orientia, Wolbachia, and a bunch of other things, okay? So essentially what you need to know is that Rickettsia is a sister, a relative of Wolbachia. And Rickettsia causes nasty diseases, many of which are spread by ticks, and some of which are spread by body lice and fleas. So Rickettsia are like the evil sister of Wolbachia. So let me take you back all the way before, even before the EAM story, long before this story even started, to 2012, a publication by Joe Gillespie and et al. A Rickettsia genome overrun by mobile genetic elements provides insight into the acquisition of genes characteristic of an obligate intracellular lifestyle. Let me show you the figure that I want you to see. The figure is figure 2. And when discussing figure 2, you'll notice that Joe starts talking about lateral gene transfer events from a plasmid of Rickettsia into Wolbachia prophages. Now look at this down here. Do you see this? This is a sequence of genes on a plasmid from Rickettsia. This is the EAM that Seth Bordenstein described later in the phage. Look at this. This green is homology, and you're seeing synteny of these genes within the EAM. What does this imply? This implies that if here's the wolfage, and here's the EAM, like I drew earlier, and the SID-A, SID-B genes are inside of the EAM, but you also see this EAM on a Rickettsia plasmid. This means that the phage, the Woe phage, stole the EAM from a Rickettsia plasmid. Boom. Why is that cool? That suggests if the SID-A and B genes are inside of the EAM and you find the EAM in Rickettsia plasmids, that suggests that other Rickettsia plasmids should have SID-A-B derivatives. Now, why is that important? Boom, tangled web drops, okay? A tangled web, origins of reproductive parasitism. This is another great paper by Joe Gillespie. And in this paper, what they describe is there's a really interesting genome called Rickettsia felis. But this is a strange Rickettsia felis that lives in a book louse. So it's not the cat flea Rickettsia felis, it's a book louse Rickettsia felis. And this Rickettsia felis has a plasmid, okay? And on that plasmid is a SIND-A and a SIND-B. What is that? This is a strange, long toxin that has nuclease domains. So it looks like SINs, but it also has a dub domain. So it looks like SIDS. So in Rickettsia, what I'm telling you is we find operons on plasmids of an A and a B with homology to SID-A, SIN-A, and homology to SID-B, SIN-B, but the B toxins are unique. The B toxins have both characteristics of the SINs and both characteristics of the dubs. What does this imply? Well, if we know the family tree of these bacteria, and there's Orientia, Rickettsia, Wolbachia, whatever was shared amongst these sisters is probably an ancestral form, okay? It's probably an ancestral form of the SID or SIN or SIND operons. And what you find is that in Rickettsia, there are SIND operons. So what's a hypothesis, and how does this connect to 2x1? Well, in this Rickettsia, it's on a plasmid. And on bacterial plasmids, you commonly find toxin antidote systems. And the reason you find toxin antidote systems on plasmids is because they are selection systems that select for this plasmid, okay? So I've drawn this before in previous lectures. If you're a bacteria and you have a plasmid, okay, and you have a toxin antidote system on that plasmid, okay, what's going to happen in that bacteria is it's going to synthesize both antidote and toxin, okay, from these two genes. Now imagine the bacteria divides. And when bacteria divide, they divide by binary fission. So the cytoplasm is shared. So this cytoplasm in this new daughter bacteria is going to have both toxins and antidotes. Now imagine a scenario where this plasmid is not replicated, is not inherited by the second daughter cell. What's going to happen? The antidote degrades and the toxin gets activated and it kills the cell. So the only cells that survive by binary fission are cells that inherit the TA plasmid. So the toxin antidote systems are ancestrally a plasmid selection system. Now it's so interesting that we find ancestral forms of SIDs and SINs, the SINDs. It's so interesting that we find those on Rickettsia plasmids, because what that suggests is it suggests that ancestrally there was a plasmid that was shared by some progenitor, a common ancestor of Wolbachia and Rickettsia. And on that plasmid was a mother toxin Cindy and Sind-A operon. And the function of this operon was to select for this plasmid. Now what happened is Wolbachia and Rickettsia diverged evolutionarily, okay? And Wolbachia somehow picked up a phage. Rickettsia don't usually have phage, okay? And somehow Wolbachia evolved CI, and there are some Rickettsia that cause CI. And what we see now is that the CI factors in Wolbachia are due to newly evolved daughters of the mother toxins, which we call SIDS and SINs. And we think that these evolve by fission of the Cindies. So if this is Sind-B, it's got nuclease, a nuclease 2, and a dub. We think that the SINs evolved by losing that C-terminal domain because the SINs look like this. And we think that the dubs evolved by loss of functionality of the nucleases. They devolved, lost their catalytic sites, but preserved dub functionality. So we think that the Sind-Bs might be some kind of an ancestral system. And the support for that is that you find these on rickettsial plasmids. And somewhere along the line, the Wophage, the Wolbachia virus, stole the EAM, which included the CI operons, and they evolved to CI from an original toxin antidote selection system. Now, in that process, in the bacteria, if you're in Rickettsia or Wolbachia or Orientia and you're a plasmid, the only function of that toxin antidote system is to select for that plasmid. So all it needs to do is kill the cells that don't inherit the plasmid. Now, what we see in CI is we see a bacteria that lives inside of an insect. And this bacteria doesn't just kill the insect, it sterilizes the insect in a very complicated system, which we call CI. And we think that this evolved to that. Now, how would this evolution happen? And how does this relate to 2x1? How does this evolution happen? Well, this process of insect sterilization is much more complex than just killing bacterial cells to select for a plasmid. That's much more complex. What has to happen is the toxin and the antidote have to be secreted outside of the bacteria. They have to get into the sperm and only kill the sperm in eggs that are not infected with other Wolbachia. So the system has to evolve in a way where it transitions to selecting for a plasmid in bacteria to selecting for the bacteria infection in a host insect. That evolution is going to be very complicated. It's evolving much more complicated functions. So in that evolution, is it possible that a 2x1 functionality has to evolve whereby A has to learn to collaborate with B to get sperm sterility and rescue rather than just cellular killing? That's what we think. So what does this mean? If we think that, if this evolutionary system is true, what does that imply? That implies that there are probably truth to both models. There's probably 2x1 systems which are highly evolved. Maybe that's the case that WMEL is one of these systems, and the 2x1 functionality possibly evolves as a crossing transition to convert from a toxin antidote system that selects for a plasmid to a toxin antidote system that selects for a bacteria. They have to learn how to collaborate together to sterilize sperm, to get to the sperm, to get to the nucleus inside of the sperm, to get out of the Wolbachia. That's a lot more functionality that has to evolve. But there are probably also still strict toxin antidote systems where B is completely sufficient for toxicity, like we observed in the SynB system in Hongley's paper, and like we might observe by studying these more ancestral versions that are in Rickettsia and Orientia. So the great question in CI of TA versus 2x1, I don't think it's one or the other, and I don't think they're mutually exclusive. I think they both exist. And it all depends on your interpretation of 2x1. If your original interpretation of 2x1 is that there are two toxins and the rescue factor is unknown, that's incorrect. But that's not what they're positing any longer. They were positing that originally. That's no longer what they're positing. If the posit of 2x1 is that the two proteins collaborate, I got no problem with that. Because I would expect that that collaboration is the functional means of evolving from a plasmid selection system to a bacteria selection system in a host insect. That is a great C that has to be crossed, and a lot of functionality has to be evolved into that system for the Wolbachia CI to convert from toxin antidote to strict CI. So let's continue the discussion even more, and let's pose the question, are these CIFs phage genes? And let's try to answer this question with a follow-up of the last discussion about the evolution of the CIFs. So the last way that I described it was that there was some progenitor, and in that progenitor bacterial cell there was a plasmid that had toxin antidote system, and that progenitor, that common ancestor, gave rise to Wolbachia, Rickettsia, and Orientia, etc. Now, if that were true, you would expect to see these genes in a lot of the Wolbachia, a lot of the Rickettsia, and a lot of the Orientia. And that's not what we observe. We don't observe these genes everywhere, okay? We find them, but not everywhere. So this is probably not quite correct. And let me posit what's a favored hypothesis by Joe Gillespie, which I agree with, which is it's more likely that these distinct groups picked up these operons, acquired them through mutually independent events throughout evolutionary history, but it's still probably true that they could have picked these up from some plasmid source before these operons invaded into Wolbachia. So I think that that discussion needs to be caveated and cautioned by how you speak about it, and it's probably more likely that there are independent acquisitions of these CI operons. So what does this mean? What does this mean that there are bacteria in far different clades, Wolbachia, Rickettsia, Orientia, and they're able to pick up sporadically these genes? What does that mean? It means that it means that the CIFs move around. It means that they jump, because they have to somehow jump into Orientia, they have to somehow jump into Rickettsia, and they have to somehow get carried into Wolbachia. Okay, so what was previously posited was that these CIFs move around. I'll put a question mark here. Do they move around in the phage? And that comes into the EAM. Does a phage carry these genes around? Well, this hypothesis works for Wolbachia. It's probably true that a phage, a woe phage, that had these operons at some point invaded Wolbachia, distributed these genes, and kind of colonized a whole bunch of different Wolbachia and deposited these. That's probably true. But that does not work for the Rickettsia and the Orientia, because the Rickettsia and the Orientia bacteria do not have phage. And they don't have scars of phage, okay? So when a phage genome integrates and maybe somehow gets pseudogenized, you can see the scars. You can see the relics of that dead phage there. Orientia and Rickettsia do not have scars of pseudogenized phages. But what they do have an abundance of is transposons and insertion elements. And these are what are jumping genes that are sometimes known to carry genes with them. So that's where this paper comes in, this new paper by Brandon Cooper, Michael Torelli, Dan Vanderpool, William Connor, and Daniel Matute. Wolbachia acquisition by Drosophila yacuba clade hosts and transfer of incompatibility loci between distantly related Wolbachia. So this was an awesome genetics paper. It came out in preprint, and that's where I originally saw the data. And it just had fantastic data demonstrating what would point to the jumping of these Wolbachia genes. So let me go right to that figure, which is this figure 4, okay? And hopefully I won't botch the explanation of this, but the y-axis here, what you're looking at on the y-axis, those spikes, that is how far it is away with respect to homology or relatedness compared to WPIP, okay? So if you see these big spikes, that means that region, it keeps popping up, that region of the genome is far away, If you see a spike, that means it's far distantly related to WPIP. If you see the lines go closer and closer to the 0 point of the y-axis, like right in this region here where you see syn A, syn B, that means it's getting closer and closer to WPIP homology. So now let me explain this. Let me translate this here in an easier way to think about it if you haven't read this paper yet, which you should. So essentially what they're looking at is they're looking at this region of a woe phage. Okay, and this region of a woe phage has two different operons, okay? It's got some SIDs, and it's got some SINs. And one of the really unique things about this paper is that these SIDs look like SIDs from WMEL. So they're very, very closely related to WMEL. And let's say WMEL is over here in a tree. Okay. These SINs are really, 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 really close to WPIP, okay? Which is like over here, more far away. So right next to each other are stacked two different operons, and the rest of the phage and the rest of the entire genome looks like WMEL. But in here, in this little region, which encodes the SINs, the syn A and syn B, this region looks like it's from Mars, okay? This region looks like it's from WPIP, which is a far off planet compared to the rest of the genome. So somehow, this little piece of extremely divergent DNA has somehow popped in, okay? And so one good hypothesis for how this happens is that jumping genes, mobile elements within the genome, carry these, and they can pop around and move, okay? So the association with the woe phage for movement might not be the only way that these things move, and it might not even be the main way in which these things move. And so if you ask the question, are these genes phage genes? as posited in the title of the Nature paper, prophage Woe genes recapitulate and enhance Wolbachia-induced cytoplasmic incompatibility. Are they phage genes? Are they phage genes? Because if they were phage genes, that would imply that they were derived from a virus and their function was for that virus. That is not what we see. What we see is that the phage probably stole them from a toxin antidote operon. The phage probably stole them from a bacterial plasmid, in which case they were bacterial genes or plasmid genes. So no, they're not phage genes, okay? Or in another scenario, they jumped into a phage from retrotransposon or mobile element activity. And in that case, they're not phage genes. They're other genes or selfish genetic elements jumping into the phage. So no, they're probably not phage genes. Which is funny, because I was criticized for, in my title, in the Nature Microbiology paper, of calling them Wolbachia genes. And I was told, no, they're not Wolbachia genes, they're phage genes. Well, no, they're probably not phage genes. Okay, so at the end of this long rant, where do we stand? What do we know to be true? Okay, so let's talk about the nomenclature. I think the best compromise and actually the most useful form of the nomenclature is CIFs. The term CIF, CI-inducing factor, use it as a hierarchy describing all sort of possible factors that are important for CI. And within that, there is an important distinguishing dichotomy in that there are SIDs and there are SINs. And actually, we know now that there are SINDs, which have both nuclease and dubs, okay? And it's very, very helpful in the lab. When you're discussing these, you cannot just say, oh, this is CIF, this is CIF, this is CIF, this is CIF. It just doesn't work. It's not utilitarian. It doesn't work, okay? So what's very useful is to say SID, SIN, SIND when you need to talk about the biochemical specifics of what you're talking about. And CIF is sort of like a broader hierarchy. If you're saying, like, we're talking about all the CIFs here. We're talking about the ancestry of all the CIFs. That's actually a useful term, so I don't mind using that. I think that's helpful. That's the way that I look at it. Where do we stand with toxin antidote versus 2x1? Okay, well, toxin antidote, if you've watched the lecture now this almost 2 hours, probably more than 2 hours, we know that toxin antidote is true, okay? A is antidote, everybody agrees. B is toxin. Everyone agrees, okay? And within that, toxin antidote is always true. Within that, there's a subset specification of a 2x1 model, whereby in the case of induction, A might collaborate with B. And we talked about how that process might evolve in the transition from a classical plasmid selecting toxin antidote system to insect sterilizing CI system, which has to become more complicated. That 2x1 functionality could evolve through that. But the 2x1 is contextually true, okay? It seems to be the case for the W-mel operon, but it does not seem to be the case for all operons, okay? So 2x1 is contextually true as a subset model within the TA framework, which is always true. And if anything I said you felt like I didn't go into enough detail, you can read about almost everything that I've talked about here in this review of ours, the toxin antidote model of cytoplasmic incompatibility, genetics and evolutionary implications. This is published in Trends in Genetics. And this is a great review. We spent a lot of time thinking about it, and it summarizes a lot of what I've just talked about. So all this is to help people in the field understand the TA versus 2-by-1 discussion. And I understand that within the field, there's maybe about 5 people on the entire planet who care about this, okay? Maybe there's only 5 people who are interested in this discussion. But for those people, that's what I think.